Hi everyone. In today's tutorial, we will go through the entire pipeline of short read alignment. Here I am showing the high level overview of individual sections of this pipeline. At first, we will perform the trimming of low quality bases using Trimomatic. We need to provide the raw FASTQ files as input, which will generate a trimmed FASTQ file. Trimming of locality bases and adapters are prerequisites for many downstream analysis of NGS data. I have made another video on how to use Trimomatic to trim the low quality bases as well as adapter sequences. Please check this video to trim the reads using Trimomatic. And in my previous video, I have shown how to align short reads into the reference genome. For that, there are two steps. At first, we need to index the reference genome using BWA. For that, we need to provide the reference genome as an input, which will generate index files. In the next step, we will use BWA mem to align the reads to the reference genome. For that, the inputs will be trimmed FASTQ files as well as index files. After alignment, BWA mem will generate a SEM file. In today's tutorial, we will cover most of the downstream processing of a SEM file. When we have a SEM file after alignment, we will convert this SEM file into the binary BAM file using SEMtools view. The input will be the SEM, output will be a BAM file. Next, we will sort this BAM file using SEMtools sort by coordinates. And this will generate another BAM file which is sorted. And then the sorted BAM file will be used to mark duplicate reads using Picard. Picard will add a flag of duplicate reads in the sorted BAM file and which will generate another BAM file which is duplicate marked. So this is the final output of this alignment process. The duplicate marked BAM file is already sorted and also there is a flag for duplicate reads. We can use this BAM file in most of the downstream analysis. For example, we can use this duplicate marked BAM file to generate the statistics of the alignment using SEMtools flagstat. It will take a duplicate marked BAM file as an input, which will generate the flagstat report. Also, we can use this marked BAM file to visualize the alignment using IGV. In my next tutorial, I will show how we can use IGV to visualize the alignment and save the results into different image file formats. So please check my next video on alignment visualization. Let's go to the coding section. You can find all my codes on my website. At first, we will install the dependencies. We will install Picard from Anaconda. Yes. Then we will install SEMtools. Installation is successful. Let's take a look. Picard is installed. Also, SEMtool is installed. Right now, in my current directory, there is a SEM file. We have generated this SEM file by aligning the reads using BWA. I have shown these steps in my previous tutorial. So now we will start from the SEM file. At first, we will convert the SEM file into a BAM file using SEMtools. SEMtools is a versatile tool that is used to work with the SEM file or the BAM files. I have made a separate tutorial on SEMtools. So today we will use SEMtools view to convert SEM file into a BAM file. -s is indicating this is a SEM file that will be used as an input, and -b that is telling SEMtools to convert it into a BAM file. Now we will provide this SAM file, and output will be a BAM file. So using this command, we are converting this SAM file and writing this output as a BAM file. BAM file is the binary version of SAM file. I'm pressing enter. Let's take a look. A BAM file has been created. The file size is much smaller than the SAM file because this is a compressed binary file of the SAM file. Now we will work on this BAM file. We will sort this BAM file using coordinates. For that, we will use sort command of SAMtools. We will provide this BAM file, and as an output, we will save the sorted.bam. And this will sort the BAM file by coordinates. Now we can take a look into the BAM file, whether it is sorted by coordinates or not. We can use SAMtools view to open a BAM file. Let's look into the header. The fourth column in the BAM file is the coordinate position. So here 1, 2, 3, 4. Yes, reads are sorted by coordinate. 1, 4, 5, 7. These are coordinate sorted. If we look into the unsorted BAM file, this will look like that. This is starting with 3532 position and 95. These are not sorted by coordinates. So using SAMtools view, we can actually open this BAM file to look into the details. Now we will use SAMtools flagstat to generate the statistics of the sorted BAM file. So this is generating all the statistics of this BAM file. Number of quality passed reads, quality failed reads, it's 0 here, and duplicates. We can see there is no duplicate. So duplicate numbers are 0. And also it's telling that number of mapped read is 96.6%. So the reason is that it's showing 0 because we did not put any flag in this BAM file to indicate the duplicate reads. So we need to add a flag so that SAMtools flagstat knows there is a duplicate read or not. Although flagstat is reporting 0 duplicate for this file, it may contain duplicates. To add flags for duplicates in the BAM file, we need to use Picard. Picard has some specific requirements to mark duplicates, such as we need to specify the read group information in the BAM file. Let's take a look whether our BAM file contains any read group information or not. If not, we need to add this. We can use this SAMtools view command to print the headers and the read groups. So in our case, there is no read group information. So now we will add read group information using Picard tool. So that is a command how we can add read group information. I will provide this command into my website. I am copying and pasting this command to add specific read group information into this BAM file. Now there is a new BAM file with read group information. Yes, now it is printing the read group information. So we can use this BAM file to mark duplicate reads using Picard. So the command will be Picard mark duplicates As an input, we will provide this BAM file. As an output, read group dupe marked, and we need to specify a matrix file that will print the statistics of this duplicate rate. Let's run this. Picard is running, marking the duplicates. Let's take a look. Yes, it has created a BAM file named duplicate marked BAM. So let's generate the statistics. If we look into this row, now it's marking the duplicates. It is telling there are more than 400k duplicate reads within this library. So before we marked the duplicates, we could not identify the number of duplicates. But using these two steps, adding the read group information as well as running via Picard mark duplicates, we can add a flag for duplicate reads, and we can also generate the statistics for the number of duplicates in the library. Finally, there is one step which is required for some downstream analysis is to index the BAM file. We can use SAM tools index and then the BAM file name. It will create a index file with .by. In most downstream analysis, we need to create the index of the BAM file, which is .by file. To summarize, in these two tutorials, we have learned about the pipeline, how to align short reads. The first step is to trim the low quality bases or the adapters, then use BWA to align the reads into the reference. Then we converted SAM file into the binary BAM file. We have sorted the BAM file by coordinates, and we used Picard to mark the duplicate reads. Also, we have generated the stats using SAM tools flagstat. This is a general pipeline. That pipeline generates the BAM file after alignment that could be used for most of the downstream analysis. The codes and the commands that are used in those tutorials can be found on my website. Please try yourself to reproduce those results and use those skills in your own bioinformatics analysis. See you in the next tutorial.